everyone, welcome to Biology with MJ. This is MJ, your enthusiastic guide. So whether you are a NEET aspirant or simply a biology lover, this video is filled with valuable insights and knowledge. Okay, so stay tuned till the end of the video, and if you haven't already, please subscribe to my YouTube channel and press the bell icon so that you never miss an update. Remember to like, comment and share this video with your friends. Okay, so let's get to the video.
Hello students. So today we are going to be talking about kingdom Monera from the chapter Biological Classification. This is the lecture number 2 of this chapter. For lecture number 1, you can visit the following link. Okay. So what are the members of kingdom Monera? Kingdom Monera comprises of bacteria. So bacteria are the sole members or the only members of kingdom Monera. It is the kingdom of bacteria. If we talk about general features of bacteria, bacteria are found everywhere. Okay, they occupy different varieties of habitats. They can live in extreme habitats. They can live as parasites. Okay, inside living organisms, they can live as parasites. They are also found in soil, in water, in the intestines. Okay, they are living as parasites. So they can live in extreme habitats, they can live as parasites, and in general they are found everywhere. Okay. Now bacteria are of different shapes. Okay. The spherical shaped bacteria are known as cocci. Okay. Cocci is the plural term. The singular term is coccus. Okay. The bacteria which are rod shaped like these are known as bacillus. The plural is bacilli. Okay, the bacteria which have a spiral shape like this are known as spirillum. Okay, and the plural is spirilla. Okay. And the bacteria which are comma shaped are known as vibrium. The plural is vibrio. So all these represent the various shapes of bacteria. Coccus, bacillus, spirillum, vibrium. These represent the different shapes of bacteria. Okay, which are spherical, rod shaped, spiral and comma shaped. Now bacteria have very simple structure. Okay, they have the simple prokaryotic cellular organization. Okay, bacteria are what? Prokaryotes, right? So they have a simple prokaryotic cellular organization. Okay, they have cell membrane. Okay, there is this outer membrane, inner membrane, this periplasmic space, cell wall is present, cytoplasm is there, ribosomes are present, plasmid, chromosomes, mesosomes, pilus, capsule. So they have a very simple structure, but their behavior, if we talk about, it is very complex. Okay, so now let's come to their behavior. Okay, they show extensive metabolic diversity. Okay, metabolic diversity in the sense of their mode of nutrition. They have both autotrophic mode of nutrition and heterotrophic mode of nutrition. Depending upon the bacterial types, different kinds of mode of nutrition exist. Okay, kingdom Monera, the members of kingdom Monera show, that is bacteria, show different kinds of modes of nutrition. There is not a single mode of nutrition that is only specific to them. They show autotrophic mode of nutrition also, heterotrophic mode of nutrition also. In autotrophic mode of nutrition, it could be autotrophic photosynthetic. Okay, it could be chemosynthetic autotrophic. It could be, if we talk about heterotrophic mode of nutrition, it could be either parasitic, it could be symbiotic, it could be saprophytic. Okay, what is photosynthetic? When they use the solar energy in order to synthesize their own food. That is photosynthetic mode of nutrition. Photo means light. So when an organism uses light, okay, that is the solar energy, the sun energy, in order to make their own food, that is known as the method of photosynthesis. Okay. and certain pigments also need to be present in order to carry out photosynthesis. Those pigments are known as chlorophyll. Okay. If we talk about chemosynthetic autotrophic bacteria, what are those? Now, photo is light energy. Chemo is chemical energy. So what happens in chemosynthetic autotrophic bacteria? They oxidize various inorganic substances, for example, nitrates, nitrites, and ammonia, and use the released energy. Okay, after oxidizing these substances, these inorganic substances, the energy that is released, they use it for the production of ATP. Okay, because when the organism will oxidize these inorganic substances, certain bonds will break, okay, which will result in the release of energy, and this energy they are using for their ATP production. Okay, so they play, chemosynthetic autotrophic bacteria, they play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. So this is both of these points are very important. If we talk about heterotrophic bacteria, they could be parasitic. Okay, which parasitic bacteria are what? Which feed or which live inside the other organisms or on the other organisms and obtain nutrition from them. Okay, they could be symbiotic. They could be symbiotic means living in a mutually beneficial association. Okay, कि दोनों organisms को ही एक दूसरे से फायदा हो रहा है. तो bacteria can also exist in symbiotic associations. They could be saprophytic also. Saprophytic means that they feed on dead and decaying organic matter in order to obtain their nutrition. So these are the various modes of nutrition of bacteria. Heterotrophic bacteria are the most abundant bacteria. Okay, if in mode of nutrition also, heterotrophic bacteria are more abundant than autotrophic bacteria. And majority of the heterotrophic bacteria are important decomposers. Okay, they are very important decomposers. They break down the dead and decaying organic matter. They are useful for humans also. Okay, for example, in order to make curd from milk, lactobacillus is used. Okay, in production of certain antibiotics, bacteria are used. So bacteria are useful. They impact a lot of human affairs. Okay, but we cannot ignore the fact that some of these are pathogenic as well. Okay, they can cause diseases in farm animals, in pets, in our crops. Okay, in humans, they can cause diseases also. Some of the common diseases that are caused by bacteria are tetanus, cholera, citrus canker, typhoid. Okay, so heterotrophic bacteria are some of these are pathogenic also, and they cause various diseases like these. So I would like to pause here, students, because I want you to follow me on Instagram, Facebook, and Telegram. Okay, so that you do not miss any update, and I post a lot of content on Instagram. Okay, so I wish to see you there. Okay, so do follow me on Instagram, Facebook, and Telegram, and do not forget to subscribe to my YouTube channel. Okay, let's move forward. Now, if we talk about reproduction in bacteria, it is of two kinds. Okay, bacteria reproduce asexually also, they can reproduce sexually also. Under asexual reproduction, they can reproduce by binary fission or by sporulation. Now, binary fission is the main method of reproduction that is seen in bacteria. Okay, it takes place under favorable conditions. But if the conditions get unfavorable, okay, for example, if the bacteria is present in an environment where moisture or water is scarce, okay, so under conditions of desiccation, okay, under conditions of starvation where nutrients are absent, these are unfavorable conditions. So in these conditions, bacteria undergo the process of sporulation, which can also be known as endospore formation. Okay, we'll talk about both of these methods soon. Sexual reproduction is of primitive kind in bacteria. Okay? It is either by conjugation, by transformation, or transduction. Now, what do we mean by primitive type of sexual reproduction? Now, in eukaryotic organisms, what happens? Eukaryotic organisms and in complex organisms also, gametes are produced. They fuse in the process of sexual reproduction. Male gamete fuses with the female gamete. Okay? But what happens in bacteria is that genetic material is getting transferred from one bacteria to another either via of these three methods. Okay, इनमें से किसी भी एक method के through bacteria, when they transfer their genetic material to another bacteria, that process is known as genetic information transfer. Okay, which leads to genetic recombination. Okay, because जो bacteria जो डोनर बैक्टीरिया का डीएनए रिसीव कर रहा है, जब वो उस डीएनए को अपने जीनोम में इंटीग्रेट करेगा तो रिकॉम्बिनेशन हो जाएगी ना. ओके, एक नया जीनोम बन गया ना, बिकॉज इट हैज न्यू डीएनए फ्रैगमेंट्स इन इट नाउ. सो ये जो मेथड है ऑफ जेनेटिक रिकॉम्बिनेशन, ये आइदर कॉन्जुगेशन से होगा, या तो ट्रांसफॉर्मेशन से होगा, या ट्रांसडक्शन से होगा. इसमें ये प्रिमिटिव टाइप का मेथड है क्योंकि यहाँ पे कोई गेमीट्स फॉर्मेशन नहीं हो रही है. ओके, सो sexual reproduction is happening, but not via the formation of gametes. The genetic material is getting transferred. In that sense, sexual reproduction is happening. Okay. Now, talking about binary fission, the parent bacterial cell will divide into two daughter cells. This is the process of binary fission. Okay. The prokaryotic parent cell initiates replication of its genetic material. Okay. A copy of cell's DNA is created. Cell elongates and the cross wall forms and two daughter cells are formed as a result. This is the diagram from NCERT. Okay, as you can see, a bacterium is dividing into two daughter bacterial cells. This is the method of sporulation or endospore formation. Okay, a single endospore forms inside the bacterial cell. Okay, and under favorable conditions, it will germinate in order to form a new bacterium. And endospore formation से bacteria can undergo long periods of dormancy. एक dormant inactive state में रह सकते हैं, protected रह सकते हैं. जैसे ही conditions favorable होंगी, they'll germinate. Okay, a new bacteria will grow. Now, if we talk about conjugation, conjugation is the process in which the genetic material from one bacterium is transferred to another bacterium through direct contact. As you can see, here a conjugation pilus is formed. So, direct contact के through the genetic material is getting transferred between the two bacterium. So, this process is conjugation. Okay. If we talk about transformation, what happens in transformation? The direct contact is not happening in this. The bacterial cell is directly taking up DNA, okay, inside it from its environment. Directly for वो जो DNA के material को वो the cell is taking in. Okay, जो donor DNA है, donor bacteria ने release कर दिया वो environment में. यहाँ पे कोई direct contact नहीं हो रहा है इन दोनों bacterium के बीच में. ये जो donor DNA है, फिर ये जो recipient bacteria है जो कि उसको receive करेगा, उसने इसको receive करा और अपने आप के अंदर integrate कर लिया, तो as a result ये transform हो गया. इस प्रोसेस को ट्रांसफॉर्मेशन बोल रहे हैं. अब इन दोनों मेथड्स में देयर इज वन डिफरेंस, दैट इज हियर कॉन्जुगेशन पाइलस इज बीइंग फॉर्म्ड, दैट इज डायरेक्ट कॉन्टैक्ट इज हैपनिंग, whereas here no direct contact is happening. The donor bacterium has released the DNA, that is the donor DNA, and the recipient bacterium has taken it up from the environment from where from the place where the donor bacteria had released it. Okay, so this process is transformation. Transduction में क्या होता है? In transduction, bacteriophages are involved. Bacteriophages are viruses that infect bacteria. इनको हम क्या बोलते हैं? Bacteriophages. So what do bacteriophages do? They take up the genetic material from the donor bacteria, and when they integrate it within themselves, okay, when its new copies are being created, see, this virus, this bacteriophage had infected this bacteria. So when it will create its copies, this bacterial DNA is getting divided and is being integrated into the newly formed bacteriophages. Now when this bacteria, after the lysis of this bacterial cell will die, and this bacteriophage will go and infect a healthy bacterium, a new bacterium, and will release its genetic material inside that bacterium. Okay, so as a result, एक बैक्टीरियम का डीएनए दूसरे बैक्टीरियम में चला गया, डोनर बैक्टीरियम का डीएनए रेसिपिएंट बैक्टीरियम में चला गया, and this process is called as transduction, and it is happening via bacteriophages. Okay, so this was about transduction. Now kingdom Monera is divided into two groups, that is Archaebacteria and Eubacteria. Now this is as per the 5 kingdom classification. Okay, as per the 6 kingdom classification, the kingdom Monera was split into 2 kingdoms, which was Archaebacteria and Eubacteria, under 2 separate domains, which were bacteria, under which Eubacteria was kept, and under the domain Archaea, under which the Archaebacteria were kept. Okay, so in the domain bacteria, Eubacteria were present, kingdom Eubacteria was present, and in the domain Archaea, the kingdom Archaebacteria was present. But as per the 5 kingdom classification, the kingdom Monera is divided into 2 groups, which is Archaebacteria, which are the primitive bacteria, okay, and Eubacteria, which are the true bacteria. Now, this is the structure of Archaebacteria, okay, a simple bacterial prokaryotic organization is present, okay, a flagellum, a cell wall, plasma membrane, cytoplasm, ribosomes, nucleoid, pilus, capsule. Now, Archaebacteria are special. They can live in some of the most extreme conditions. Okay, they are of 3 types. They are either halophiles. Halophiles are what? Which live in salty areas. The bacteria which can live and survive in salty areas are halophiles. Thermoacidophiles, which can survive in hot springs, okay, where temperature is very high. Methanogens are those bacteria which live in marshy areas or in the guts of ruminant animals, for example cows, buffaloes, okay, and these kind of bacteria are responsible for the production of the methane gas, जिसको हम biogas भी बोलते हैं, okay, from the dung of these animals. ठीक है, they live in the gut of these ruminant animals and they are responsible for the production of methane or biogas from the dung of these animals. These group of bacteria are known as methanogens. Okay. How do they survive in such extreme conditions? What makes Archaebacteria so special? So number 1 is that their cell wall lacks peptidoglycan. Okay. Contrary to the Eubacteria, their cell wall lacks peptidoglycan. Eubacteria में peptidoglycan is present, but in Archaebacteria, peptidoglycan is absent in their cell wall. Then cell membrane also. Cell membrane of Archaebacteria has branched chain ether-linked lipids. Okay, whereas the cell membrane of Eubacteria have straight chain, that is unbranched ester-linked lipids. Okay, as you can see in this picture, this is the phospholipid from Archaea and this is the phospholipid from Eubacteria. So Archaea में क्या है? Archaebacteria में ether linkage है और Eubacteria में है ester linkage. ओके, Eubacteria में क्या होता है? Phospholipid bilayer present है, ये phosphate group है and this is the fatty acid tail. So एक लेयर ये है, एक लेयर ये है, whereas Archaebacteria में a single layer is present. Okay, as you can see, a single layer is present, a phospholipid monolayer is present. So ये जो features हैं, ये branch chain ether linked का lipids का होना, it decreases the membrane fluidity. Okay, under extreme conditions इससे बैक्टीरिया का जो स्ट्रक्चर है वो स्टेबल रहता है, बिकॉज फ्लूइडिटी जो है वो डिक्रीज हो जाती है। ब्रांच चेन होने की वजह से लिपिड्स का जो फ्री मूवमेंट है वो स्टॉप हो जाता है। ओके वो इतनी फ्रीली मूव नहीं कर पाते बिकॉज उनकी ब्रांच चेन्स प्रेजेंट है एंड ईथर लिंक्ड लिपिड्स है। ईथर के बॉन्ड होने की वजह से दे आर ऑल्सो रेजिस्टेंट टू डिग्रेडेशन बाय सर्टेन एंजाइम्स, जो कि हार्श कंडीशंस में काफी सारे इन्वायरमेंटल फैक्टर्स होंगे जो कि बैक्टीरिया के स्ट्रक्चर को डिसरप्ट कर सकते हैं, बट ईथर बॉन्डिंग होने की वजह से वो उससे प्रोटेक्टेड रहते हैं बिकॉज इट इज वेरी resistant to enzymatic action or the external environmental influences because it is a very strong bond. ओके एंड ब्रांच चेन होने की वजह से जो लिपिड्स का फ्री मूवमेंट है वो स्टॉप हो जाता है, जिसकी वजह से फ्लूइडिटी जो है वो डिक्रीज हो जाती है एंड मेम्ब्रेन का जो स्ट्रक्चर है वो रिजिड रहता है। इतनी इजीली वो डिसरप्ट या डिसिपेट नहीं हो सकती अंडर एक्सट्रीम कंडीशंस। ओके नाउ यूबैक्टीरिया की बात करें तो यूबैक्टीरिया आर मोर अबंडेंट, they have a rigid cell wall, they are more abundant than Archaebacteria. They have a rigid cell wall and they also have a flagellum if they are motile. Okay, not always, but if they are motile they'll always have a flagellum. They have ester linked lipids and their cell walls have peptidoglycan. So peptidoglycan is present and ester linked lipids are present. Okay, so this is the structure of eubacteria. It will look very similar to that of an archaebacteria. Okay, all the same components are present: cytoplasm, nucleoid, DNA, ribosomes, plasma membrane, cell wall, capsule, flagellum, pilus. So the only difference between eubacteria and archaebacteria mainly lies in where they live, okay, and then in their cell membrane structure, okay, their cell wall structure. Okay, because ester linked lipids are present in eubacteria, ether linked lipids are present in archaebacteria. Peptidoglycan is present in the cell wall of eubacteria, but it is absent in the cell wall of archaebacteria. Now talking about cyanobacteria, which is also known as blue-green algae. Now cyanobacteria are a type of eubacteria. Okay, they are one of the groups that fall under eubacteria. They're also known as blue-green algae because earlier they were put in algae, but they are not algae. Then they were put into the right place, which is under the eubacteria category. Okay, but ये जो term है ये तब से use वैसे ही की जा रही है. Now they have chlorophyll A in them. They are photosynthetic autotrophs. Okay, they can synthesize their own food by using chlorophyll and the energy of sunlight. They are unicellular. Okay, they can either live in colonial or filamentous forms. Okay, so this is a filamentous form of nostoc, which is a cyanobacteria. They can either be fresh water, marine, that is sea water में रह सकते हैं वो, या फिर वो terrestrial हो सकते हैं, which is living on the land. So this is the structure of nostoc. So, it is surrounded by a mucilaginous sheath. Okay, so cyanobacteria are covered by a gelatinous or a mucilaginous sheath like these. Certain of the cyanobacteria like nostoc and annabena, they also have a heterocyst, which is a specialized cells that is responsible for fixing atmospheric nitrogen. Okay, it is responsible for the nitrogen fixation. Okay, so this was about cyanobacteria. They form blooms in polluted water bodies. Cyanobacteria, okay, they can often grow in polluted water bodies and form blooms like this. So ये जो green color के सारे structures हैं, these are cyanobacteria. and ये blue green दिख रहे हैं because तभी इनका नाम पड़ा है हमें आ रहा blue green algae. Okay, now coming to mycoplasma. Now mycoplasma is also one of the groups that falls under eubacteria only. Okay, but it has some major differences from the other members of the eubacteria. Okay, क्योंकि they completely lack a cell wall. अभी हमने पढ़ा कि eubacteria में cell wall is there, but ये mycoplasma एक special और एक exceptional type का group है तो ये eubacteria के अंडर ही आता है, but it completely lacks a cell wall. Okay, इसका जो structure है it is just surrounded by a cell membrane, जिसकी वजह से it can change its shape, जिसको हम प्लियोमॉर्फिज्म भी बोलते हैं, बिकॉज इट इज जस्ट सराउंडेड बाय अ प्लाज्मा मेम्ब्रेन. तो इसकी स्ट्रक्चर में कोई रिजिडिटी नहीं है, इट कैन change its shape, its morphology. Mycoplasma are the smallest living cells known. सबसे छोटे living cells जो known हैं, these are known as mycoplasmas and they can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. They are pathogenic also. So this is the structure of a mycoplasma. So lipoproteins are present, DNA is present, ribosome is present, RNA, metabolites. Okay, so this is just a general structure of a mycoplasma. Outer plasma membrane and inside is the cytoplasmic area. Now coming to PYQs. Question number 1 is from NEET 2017. Which among the following are the smallest living cells known without a definite cell wall? That is, cell wall is absent. Pathogenic to plants as well as animals and can survive without oxygen. Okay, so is it Bacillus, Pseudomonas, Mycoplasma, yeah, Nostoc? We have just studied about Mycoplasma and all of these points match the description of Mycoplasma. So the correct answer is option number C. The primitive prokaryotes responsible for the production of biogas from the dung of ruminant animals include the halophiles, thermoacidophiles, methanogens या eubacteria. So primitive prokaryotes हमारे क्या होते हैं? Archaebacteria, and Archaebacteria are of 3 types. These are halophiles, thermoacidophiles and methanogens. So methanogens में हमने पढ़ा था that methanogens are responsible for the production of biogas from the dung of ruminant animals, for example cows and buffaloes की. Okay, so the correct answer is option number C. Methanogens belong to, we have just studied again, methanogens belong to Archaebacteria. So आप देख सकते हो methanogens पे कितने repeated questions आ रहे हैं. So this methanogens are very important. Okay, so अच्छे से इसको याद रखिएगा. Now coming to our AIPMT 2014 question, Archaebacteria differ from Eubacteria in cell membrane structure, mode of nutrition, cell shape या mode of reproduction. हमने अभी पढ़ा था कि Archaebacteria and Eubacteria में everything is similar. Okay, there is only one major difference, that is उनका जो cell membrane है and जो उनका cell wall है उनके composition में फर्क है बस. So हमारे options में इन दोनों में से कौन सा point आ रहा है? Option number 1. Their cell membrane structure differs from each other because in Archaebacteria ether-linked lipids are present, whereas in Eubacteria ester-linked lipids are present. And Archaebacteria में जो lipids हैं वो है हमारे branched chain lipids, whereas in Eubacteria the lipids are straight chain lipids or unbranched lipids. Okay, so the correct answer is option number A. So that was all for today's video, students. Hope you enjoyed the video.
Thank you for watching this video. If you have any feedback and suggestions, or if you found something useful, do let me know in the comments below, and like this video, definitely share it with your friends and subscribe to my channel. See you in the next video. Bye bye.